Good evening, all. I hope I am audible. Can anyone confirm if I am audible or not? Can anyone confirm if I am audible or not? Okay. So we will start with our session. I will share my screen. I hope my slides are visible. Okay, I think they are little blurry, so I just change the network. Yeah, it's better now. Uh, Again, good evening all. So we will start with our problem solving session for week 6. The topic we will be covering today are variant calling, calling SNP and SNVs and indel, hands-on analysis, variant calling, VCF files and variant annotation. So this course is taken by Professor Riddhimandhar. He is a professor in IIT Kharagpur in Department of Biotechnology. So I'll just quickly, briefly introduce myself. I am Pooja Yadav. I am a PhD scholar as well as a PMRF fellow in IIT Bombay, uh, in Department of Biosciences and Bioengineering, and I am a PhD student in Molecular Virology lab. So we will start with our session now. Okay, till week 5, we have covered um, we have covered read data from the sequencer that we get after the sequencing. Then we did data quality control. We did quality check for the for our raw reads, and we pre-process our raw reads. uh, then we mapped those raw reads to the reference sequence, and we did the assembly. So in the 6th week, we have learned that after the assembly, we need to analyze our data. And the objective here for analyzing our data is to find the genomic variant or the mutations or SNPs, indel, SVs in the assembly file. So in the sequence which is of our interest that we have assembled with the reference sequence, now we want to see whether the sequence that we had is exactly as same of the reference sequence, or is there any change in the genes which might be responsible for any change in the overall phenotype of an organism where we are getting the genome. So for that we need to further analyze our data. So after read assembly, we got .bam file. So for the so this gene variant that we will be getting, we call it variant calling. Variant calling means that we are calling for the variants which are present in the genome of our interest. So for this, there are many steps. The variant calling is done commonly by SAM tools and BCF tools. So first, what we do, we convert the SAM file to a BAM file. SAM file is a text-based file. BAM file is a binary file. Then we sort the BAM file. Then we do the local realignment. Then we mark for the duplicates. Then we call for the base quality sequence recalibration. Then we call the variant calls. So this all these steps are done because we want to make sure that the variant that we are calling is actually the variant and not the artifact. So we do all these steps again, like sort the BAM file, realignment, mark duplicate, and then we give the this quality again for the variant that we are seeing. Then we call the variant, so that we can be sure that the variant that we are getting or the mutation that we are getting is actually the mutation, not it is because the some artifact, or it is coming from the sequencing error, or it is coming from the read duplicates. But it should be actual a variant to be it should actually be a change in the genome to call it as a variant. So to make sure, we do all these steps and then we further analyze our data. So today's lecture will be based on these topics. So we will be having some questions. So we'll go through each questions. So first, again, I would like to. I expect you to answer the question first. Then we can have the discussion on each of the questions. So we'll start with our session. We'll start with our question first. The question first, it states that which command do we use to convert a SAM file to a BAM file? A is SAMTool sort, B is SAMTool index, C SAMTool view, D is SAMTools fixmate. Please try to answer this question. I hope I am audible and my slides are visible. Okay, we have one answer A, SAMTool sort. Okay, so before sorting, so just go to this step. So before sorting the BAM file, we need to convert SAM file to a BAM file. So this question is asking which command do we use to convert a SAM file to a BAM file. One of the answer is correct, Karthik. So we have another option C, SAMTools view. Is there another answer that might be correct for this question? So we will look into it. So, okay, sorry. So let's go through this manual. I share the link into the chat box as well, so you can also look into it. So I'll just share it. Okay, so here it is written that SAMTools is a set of utilities that can manipulate alignment formats. It imports from and exports to SAM, BAM, CRM and does sorting, merging and indexing. So it is basically a tool which does all these things: sorting, merging, indexing, and allows read in any region to be retrieved swiftly. So Converting a SAM alignment file to a sorted index BAM file using a SAM tool. So this command is used, which is SAM tools view. So for converting a SAM file to a BAM file, a SAM text file to a binary file, we use the SAM tools view. So I've shared the link for this manual. You can go through it and look at all the commands, what are the commands and how they are used and when they are used. So just take two. Okay, my voice is breaking. Is it still breaking? I think we have network issue today. Is it better or is it still breaking? Okay, now. So the command for converting a SAM file to a BAM file is SAM tool views. So I have shared this link for this manual in the chat box. Just go through the manual, just take 2 minutes and try to see what are all the commands here and what they are used for. Just take 2 minutes. So SAM tool sort, it is used to sorting this alignment will allow us to create a index. So sorting a BAM file to create an index. Then there is SAM tool index command. Just go through this file manual. So the correct answer for this question would be SAM tool views. So I hope this question is clear. Okay, we'll move to our next question. Question number second. Which of the following processes are performed during the duplicate marking by SAM tools? A. Sorting reads by name. B. Fill in made coordinates and insert size field. C. Sort based on chromosome number and coordinates. D. Base recalibration. Just go through this question and try to answer this question as well. So after the local realignment, we mark the duplicates, and in this question, what is asked? So which of the following processes are performed during the duplicate marking by SAM tools? We have one answer, A and C. Are there any other answer that might be correct, including A and C? Okay, so we will look into it. So again, here we have another manual. So I'll also share the link for this in the chat box. So duplicates, what are duplicates? Duplicate reads, as we know, they are over representation of sequencing data at the location of those reads. So the reads are getting duplicated. So the same, we have a multiple duplicated of this duplication of the same read happening in our data file. So to mark the duplicates, there are a workflow which mark the duplicates. And so this SAM tool collate it um, do. I will just wait. So we have this. So this SAM tool collate it sort the read by name. So this is the workflow which is done by SAM tools to mark the duplicates in the file. So it first sort the reads by name by SAM tools collate, and then it fill in mate coordinate and insert size field by fixmate, this command, and then it sort based on chromosome number and coordinates by SAM tool sort minus o position sort. So these are basically, and then it mark the duplicates. So this is the simple workflow for marking the duplicates by SAM tools. So what it does, it sort the reads by name, fill in mate coordinates and insert size field. It sort based on chromosome number and coordinate. So I have shared this manual with you, the link for the manual. Just again go through this file and just see what are being done by this command and tools. The correct answer would be A, B and C. Just go through this manual for a while, then we can move ahead to our next question. Let's move to question number 3. Um, Question number 3 states that choose the true statements about BQSR base quality source score recalibration. Just go through this question and try to answer this question. Go through the options and try to answer this question. We have one answer, A, B and C. We have another answer, B and C. Are there any other other options that might be correct, including A, B, and C? Okay, so we will look into the answer. So let's go through this manual. I hope my screen is visible. So BQSR, it stands for Base Quality Score Recalibration. It is a data processing step that detects systematic error made by the sequencing machine when it estimates the accuracy of each base call. So when so we have actually gave the quality score for the each basis when we were pre-processing our this raw reads. So this is again why we are calculating the quality score, because here again we want to be make sure that the variant that we are calling is the actual variant and not the artifact. So it detects systematic error made by sequencing machine, which when it estimates the accuracy of each base call. So to avoid that any error that has been this any errors that is made by either sequencing machine or maybe another sort of error, so just to avoid any error we gave again the base quality score, so that we want to be sure that the variant or the change that we are detecting is the actual change and not the artifact one. So here what are the options? So it allows us to get more accurate basis quality and improve the accuracy of the variant call, which is correct. BQSR can help in improving variant calling or genotype analysis, which is also correct. It is a measure of confidence in the base call, which is also a correct. We are being it gives us more confidence about the quality or the variant that we are calling. BQSR helps to identify duplicate read present in the library. It is not correct. It does not help to identify duplicate reads. E option is this process uses known variants to mask out bases at the site of real variation to avoid counting them as error. So BQCR, it is based on machine learning. So we gave the already known data to our to the system. So then with the help of ML, so uh, it uses the known variant to mask out the bases. So it uses the known variant which is already present in the database. So to call the variant they are calling is actually the variant and not the artifact. So they use ML. So this option is also correct. This process use unknown variant to mask out known variants to mask out bases at sites. So these known variants are already present in the database. So we get the data from the database. We gave to the software and then we compare it with the variant that we are calling, whether it is actually a variant or it is the artifact. So the correct option would be for this question is A, B, C, and E.
Madam, this thing will work out only for the known variants, right? For novel variants, it might not work, right?
Yeah, it won't work out for the novel variant. For the if we are getting a novel variant, we look into other parameters. So we look into the duplicate number, we look into the read number, we look into the quality. So we look for all the combinations as well if we are
Okay.
getting a novel variant, not just the
Okay.
one.
Okay. Okay.
Yes. So I hope this question is clear. So it depends on what is your objective of what you are looking at, so we can do whatever like we would like to do. So if this question is clear, we'll move to our next question, question number 4. Which of the following options are true about BCF file format? Just go through this question and try to answer this question. So after variant calling, we get this VCF file, which is variant calling format. So in this question, it is asked what is BCF file format? What is true about BCF file format? We have one option A, C, and D. Is there any other option that might be correct for this question? Okay, so we'll look into it. So again, we'll go into the site. So the variant call format is a standard for storing variant data. So when we do this yeah, sure, I'll share the link in the chat box again. So the variant call format, it is a standard for storing variant data. So after variant calling, we store our data into a VCF file. So this originally designed for SNPs and short indels. It also worked for structural variants. It is a text file format, most likely stored in a compressed manner. It contains meta information. We will look into it. What are the meta information that are stored in the BCF file in upcoming questions? A header line and then data line, each containing information about the position in the genome. So these lines contain the informations which are present in the genomes where the variant has been called. So BCF or a binary variant call format. So variant BCF is a text file while BCF is a binary variant call format file. So it is a binary version of VCF. It keeps the same information in VCF while it is much more efficient to process, especially for many samples. The relationship between the BCF and VCF is exactly the similar as BAM and SAM. So BAM was the alignment file, the text file for the alignment, and SAM was the binary file. The SAM was the text file for the alignment, while the BAM was the binary file for the alignment. It is same for the BCF and the VCF. VCF is text file for the variant data, while BCF is the binary file. So it is much more efficient to process, especially for many samples. So BSCF file is being read by our system very easily, so it is easy for the processing of BCF file in comparison to VCF file. So the correct answer for this question would be A. It is designed to store the data in an efficient manner and compact way, due to which the file size is smaller than VCF file. C. It contains both variant and genotype information. D. It is faster to process and manipulate. So B is not correct. It is a binary file. It is not a text-based file. It stores the variant calling information. So the correct answer would be for this question are A, C, and D. We upload BCF file, not the BCF file for IGB, because there we see the texts. So if this question is clear, we'll move to our next question. Match the column in context of VCF file. So in the previous question, I have mentioned there are certain lines, meta information line, header lines, and the data lines. So these lines, they contain certain fields which give all these informations. So now we need to match that information which is present in what field. So I'll provide a link in the chat box. Just go through the link and try to answer this question. So I have provided the link in the chat box. Just go through this link and try to answer this question. Take few minute time. So so this is the VCF file. So in blue, these are all the meta informations of the file, and then this line, it is a header line, and this is the data line. So header line represent the kind of data which is present in the data line. So in all the meta information, header line as well as the data line, certain fields are present, and these some of the fields are mandatory and which give certain information about the data that we have. So this is the information and these are the fields. We need to match the information to the field. So just go through the manual and try to answer this question. have one option, D. We have, is there any other option that might be correct apart from the D? Option D is correct. So allele frequency, it comes under the field info. Total depth also come under the field info. Read depth, it comes under the field format. Q10, it is quality as well as it is a filter as in the quality score below 10 we discard them, we does not like count them as a variant, so it is sort of providing a filter to the variant calling. So it is a filter and a quality. It is a score. Q10 is a quality score as well. 5, it haplotype quality is again a format. Phasing is this genotype. Phasing comes under genotype, which means we differentiate among mother and parent haplotypes. And 7 is heterozygous, which is 1 or 0. It either heterozygous or homozygous, so 1 and 0. So when we look into the file, so this is a manual for variant call format, so you guys can look into it even after the class. So here everything is elaborately explained. So here this is the meta information, this is the header line, this is the data line, and each has certain fields. Here you can see the fields. So these, some fields are mandatory, some fields are optional. So the mandatory, like the mandatory fields are, so these are all the mandatory one, the info, filter, quality, reference ID, position, chromosome. These are all the mandatory fields, and there are certain optional fields as well. So, and here in these fields, certain informations are given, such as the information about allele frequency, depth, read depth, quality score, phasing, the chromosome number, etc. So the correct answer for this question would be D. So you can refer to this link after the class as well. So just to know more about the VCF file formats and their fields and how you can read this file. So I hope this question is clear. So we will move to our next question, question number 6. In a VCF file format, filter column has a value description of S50. What is the meaning of this value? Only 50% coverage across chromosome, only 50% coverage across allele, data is covered by less than 50% of sample, data is covered by more than 50% of sample. Please try to answer this question as well. We have one option D. So you can again refer to the manual that I have provided for the previous question. There also you will find answer for this question. So try to look this into the file and then try to answer this question. Filter field is basically tells you about the quality of the base that we are calling at the certain position, or the quality of the variant that we are calling. So look into the field of filter. We have one option which is C. Is there any other answer that might be correct apart from C? So the C option is the correct option. Data is covered by less than 50% of sample. So we will look into how. So S50. If I search, so in filter column, so it is uh, key and it equals to value. So here it is given S50. So the description, what this S50 tells, that less than 50% of sample have the data. Let's suppose we have the quality Q10 for a particular variant, and it is also given S50 for the same variant. So we say that this variant having the quality less than 0 is covered by the less than 50% of the sample that is present there. So
Actually, sample means the number of reads, ma'am.
yes, number of reads. Here
Okay.
we can say number of reads, right? So here it is also one example is given, Q10 semicolon S50. It might indicate that at this side the quality is below 10 and the number of samples with the data is below 50% of the total number of samples. So it gives that how many like the number, how many number of samples, the number of samples with the certain data is below 50% of the total sample. So the correct option for this would be the data is covered by less than 50% of the samples.
So it means that less than 50% reads in that particular portion are having
That
quality of 10.
yeah, quality of 10 or any other quality that might be assigned
Okay.
to that.
Okay. So less than 50% means it is rest 50% are having above the threshold.
Yeah, it is below the threshold. I mean, if it is less than it is present in the less than 50% of the population, so that data
Okay.
might be not good. It something tells me.
Okay. Okay, fine.
So I hope this is clear. So the correct option would be C. The data covered by less than 50% of the sample. So we will move to our next question. Question number 7. Which of the following statements are true regarding frame shift mutation? Go through this question and try to answer this question. It's a very straightforward question. We have one option A and C. Are there any other options that might be true apart from A and C? We have another answer which includes A, C, and D. So the correct option would be A and C. An insertion or deletion of a nucleotide from a DNA sequence causes a frame shift mutation, which is true. B, frame shift mutation are always silent mutations. No, the frame shift mutation are not always silent mutations. Silent mutation meaning the mutation is after the mutation, the codon is coding for the same amino acid, which is not always the case for the frame shift mutations. The C option is frame shift mutation can lead to the production of truncated protein. This is true. If due to the frame shift mutation there is um, a stop codon is coming to the frame, then there is the truncation of protein happens. The D option is frame shift mutation cannot lead to a change in the amino acid sequence of the protein. Yes, they do change for the amino acid sequence if the new codon, because of the frame shift, if the new codon is coding for a different amino acid, they change for, they lead to change in the amino acid sequence. So I just show how it is done. So let's suppose we have a sequence. Yes, A and C are correct. We have a sequence A, T, G, C, T, A, G. So we have one codon here, one codon here, T, T. And let's suppose there is a insertion A, T, A. Let's suppose there is an insertion here. So due to this insertion, the frame has been shifted by position 1. Initially we had these codon, now we have different codon. And let's say that these codons, they are coding for different amino acid. Initially this CTA was coding for amino acid 1, and this GCT is now coding for amino acid, a different amino acid from CTA. So uh, so the correct option would be A. An insertion or deletion of a nucleotide from a DNA causes a frame shift mutation. Here there has been an insertion happened, so it changed co- the codon which are present in the sequence. It has changed the frame of the codon. The frame shift mutation are always silent, which is not the case. The change that is happened due to frame shift mutation, they can code for a different amino acid. The new codon that we have can and they generally code for a different amino acid than the previous codon. And a frame shift mutation can result in the production of truncated protein, which is also true, because if due to the frame shift mutation uh, there has been the insertion of, not the insertion, there has been a change of codon to a stop codon, that in the synthesis of protein which stops in between where this stop codon is coming, so it results in the truncation of the protein. And the D option is frame shift mutation cannot lead to change in the amino acid sequence, which is also not true, because they always not code for the silent mutations. They can lead to change in the amino acid. So the correct option would be A and C. So I hope this is clear. We'll move to our next question. Question number 8. Which of the following is not a feature type in GFF format? A. Exon, CDS, gene, transcript, protein. just go through this question and try to answer this question. GFF file is a gene annotation file. So what are the features this file does not contain? We have one option which is protein. So after the assembly, we do the annotation of our genome. So during the annotation, we get one of this file .gff format file, which is for the gene annotation. It is not the genome annotation, it is the gene annotation file. So the correct option is protein. So this is the example of g.gff file. So it is a gene annotation file which have the information about the gene, the transcript mRNA, the exon, the CDS. Um, so it does not have the information of for the protein. We can get the information of the protein from the gene, but this file does not have the information about the protein sequence. This only have the information about the gene sequence, which is the DNA sequence of the gene. So, and the mRNA as well, the transcript information. So the correct answer for this question would be E, protein. Protein is not a feature type in GFF format. So if this question is clear, we'll move to our next question. What are the key feature of GTF files? A, hierarchical organization of feature. B, sequence alignment data. C, annotation of gene and transcript. D, protein sequence of gene. Please try to answer this question as well. It is similar to GFF file only. It also contains the information about the gene annotations. So try to answer this question as well. So there are two files, GFF and GTF files, which is for the gene annotation file. So all the information that are present in the GFF file also present in the GTF files. So all the information which is present in GFF file present in the GTF file, but there are it contains some additional convention specific to the gene information. We won't go into the detail, and it hierarchically organize all the feature. So here you can see, so it is organized hierarchically only. So based on the position, based on the reference number or the position, it organized in such a way. Apart from the information which is present in the GFF file, few additional information are present. So the correct option would be hierarchical organization of the features, annotation of genes and transcript. It does not contain the sequence alignment data. Also, it does not contain the protein sequences of gene because it is same as of GFF file. So the correct option would be A and C. So we'll move to our next question. Which of the following file formats can be used to represent genomic annotation? GTF file, GFF file, FASTA, BEGD, BCF. Please try to answer this question. We have already looked two files. So is there any other file which is used to represent genomic annotation apart from the two that we have looked? We have one option A, B and D. Are there another option that might be correct apart from A, B and D? The correct option is A, B and D, the GTF file, GFF file and BEGD. So we have already looked into GTF and GF. So BEGD format is also it is also used to define the coordinates of the genomic regions and feature. So how the BEGD format look? It contains the information about the chromosome number, the start chromosome, the end chromosome. So we will look into how this BEGD file looks like. So I'll share this here as well into the chat box. So this is the example of the BEGD format file. It contains the chromosome number, the chromosome start site and the end site. It can contain the another information as well, the positions, the start and the end positions, the strand information, positive strand or the negative strand, whether it is a forward sequence or the reverse sequence. So along with the GTF and GFF file, BEGD file is also used for genomic annotations. So the correct option would be A, B and D. BEGD file does not annotate the gene, it is for the genome annotation, genomic annotations. So the correct options are A, B and D. So we'll move to our next question. Question number 11. Which of the following statements are true regarding the info field or format field in a VCF file? Please go through this question, and I have shared a link with you of the manual of VCF file. So you can also refer the manual and try to answer this question. I'll again share the manual into the chat box. Just go through the manual and try to answer this question. have one option A, B, and D. Are there any other option that might be correct apart from A, B, and D? So we will look into it. So here this is the VCF file format, and here the meta information is given, and in the meta information we have info field and we have a filter field, and here we have key. So both info field and the format field, they have a key and the value. So this the first option, it states that the info field provide additional information about the variant such as allele frequency, total depth, and number of samples. So here you can see that the info field, it can provide the number of samples, the total depth, the allele frequency, ancestor allele, so SNP membership, and other information. So this option is correct. So the second option is the format field is used to describe the data types and order of the field that appear in the individual genotype columns. If we look into the filter column, we have the quality score, the the genotype information, the read depth, the haplotype information. So it contains the data type and the order of the field that appear in the individual genotype. So it is also correct, but because it is providing all the information about the genotype quality, haplotype quality. The C option is the info field can only store a single key value pair for each variant, while the format field can store multiple value for each sample, which is not correct. Both the info field and format field uses a key value pair system where the key is unique identifier and the value is the corresponding data. So here we are given with the key and what is the value for the key for both the info and the format field. So the D option is also correct. So the correct option for this question would be A, B, and D. So if so, just go through the file, the manual file, and even after the class, you can go through the manual file and look into it, how all the information are given in the VCF file and how you can read a VCF file. So this manual would be helpful in that. So we'll move to our next question. Which of the following statements are true? Uh, A. Synonymous mutation may cause changes in the protein folding. B. Non-synonymous mutation changes not only the DNA sequence but also the amino acid sequences. C. Nonsense mutations are a subset of non-synonymous mutation. D. The functional changes in the protein are only caused by the non-synonymous mutations. Please go through this question and try to answer this question. I hope I am clearly audible. So the synonymous mutation is the mutation. Okay, so after the mutation, let's suppose a codon is coding for amino acid. So synonymous after a mutation, the new codon will also code for the same amino acid. So this is the synonymous mutation, and non-synonymous mutation is the mutation which changes the amino acid sequence. So which changes the which amino acid is being synthesized. So in the non-synonymous mutation, the different amino acid is synthesized, while in the synonymous mutation, the same amino acid is synthesized. So we have one option which is B, C, and D. Are there any other option that are correct apart from B, C, and D? So we will look into it. The correct option or a subset of nonsense. Okay, the correct option are B and C. So in the non-synonymous mutation, for the option A states synonymous mutation may cause a change in the protein folding. So commonly what happens in the non-synonymous mutation, there is no change in the amino acid. So there is a chance that the majority cases there is no change in the protein folding as well, because the amino acid are the same. The B option is non-synonymous mutation changes not only the DNA sequence but also the amino acid sequence. So which is true, because during the non-synonymous mutation there is the synthesis of different amino acid. So the mutation is causing the change in the DNA sequence, and because it is a non-synonymous mutation, there is also a change in the amino acid sequence. The C option is nonsense mutation are a subset of non-synonymous mutation. So nonsense mutations are basically wait a minute. Nonsense mutation are basically where the protein sequence are being truncated, are not synthesized fully because of these mutation. So this is also a subset of or a type of non-synonymous mutation, because here the protein sequence is different as of the wild type protein sequence. So it is a subset or the type of non-synonymous mutation. For the D option, the functional changes in the protein are only caused by non-synonymous mutation, which is functional changes in the protein, which is also true. B, C, and D. I'm sorry. B, C, and D are correct. So this is a common annotation that synonymous mutations does not do any change in the protein sequences or functionality of the protein, while the non-synonymous they do change the amino acid sequence as well as their folding and their function. But there are certain regions in the DNA sequence, if the non-synonymous synonymous mutation happen, they may change the protein folding or in fact the function of the protein as well. So let's suppose there is a non-synonymous mutation happens in the regulatory region of the gene, so which actually controls the protein synthesis which actually controls the protein synthesis of the gene. So when certain, even the synonymous mutation happen in those regions, they can cause the misfolding of the protein or the functionality of the protein. So this basically goes for if the mutation is happening in the gene region of the sequence, not in the regulatory region, because for that this might not be correct. So, but for here in this class, the option would be B, C, and D. So you can read upon it when the non-synonymous mutation can cause the change in the protein functionality, and there is also a codon usage bias where during the translation process certain codon are being used for the protein synthesis more than that of the other codon when they are coding for the same amino acids. So you can read upon it.
Yeah, madam, in the assignment section which was given to us,
Yeah.
multiple options were not there.
Okay.
Okay, only one option we could correct. The question might be changed, which of the statement is not true. Then we can answer one, because all these three are correct, but we cannot answer all these three.
Okay, then you can, I mean, like, raise the query on the portal, because, like, if you can select for one, then if you select either of them, which might be true, they'll have to give marks for either of them, or you can raise the query on the portal that these options were correct, and like like choosing for only one option was there.
Right, fine.
Yeah.
Ma'am, as Karthik pointed out, that there are not multiple options for available to select. So can we choose that non-synonymous mutation changes not only the DNA sequence but also the amino acid sequences only?
You can change it because it is also a true, but another options are true. It would be better if you guys raise a, like, if you choose it right now, but also raise a query that these options were also true, but you could not select because, like, there this was not a multiple choice question for few people. Because of course you can choose
Yes, sir.
one of the either answer, but again just raise a query on the portal, so they'll also be clear about it, that people will of course be choosing different answer for this question. So I hope this question is clear. So we'll move to our next question, which is question number 13. A CDS in a GFF file is given by the sequence, which is given here. In the frame column for the feature is 2. What will be the number of amino acid from this sequence that are transcribed and translated? So just go through this question and try to answer this question. So CDS is the coding sequence of where the protein is getting synthesized. We are given with the CDS sequence, and it is given that in the frame column for the feature is 2. You need to remember what is this, what does it mean when feature has frame has 2 in the feature as a feature. So now you have to answer what is the number of amino acid that are being coded by this CDS. Has anyone got the answer for this question? So, okay, we'll look into the answer for this question. So, what this means. So, in the GFF file, so this frame column, it indicates the number of bases to skip at the beginning of the sequence before starting translation. So this number is usually 0, 1 and 2, where 0 it means translation start from the first base, 1 it means translation start from the second base, 2 it means translation start from the third base. In a question, what is given in a frame option, we have 2 as a feature. So we'll have to ignore the first 2 bases and then we'll have to include the rest of the bases for the protein synthesis. So again, try to answer this question. Just ignore the first 2 of the bases of the start and then calculate the number of amino acid that are being synthesized by the rest of the bases. We have one answer which is 6. So the correct option would be 6 only, because if we ignore the first 2 bases, we are left with the 6 codon. So 1, 2, 3, 4, 5, 6. So each codon codes for 1 amino acid. We have 6 codon. So we will have the sequence of 6 amino acid. So the correct option is 6. So we'll move to our next question. Which of the following statement are true regarding germline and somatic variants in the context of genetics? Please go through this question and try to answer this question. Germline variants can be inherited from one or both parents, while somatic variation are acquired during an individual's lifetime. Somatic variants are always pathogenic, while germline variants are always benign. Somatic variants can be passed on to offspring, while germline variants cannot be inherited. Phasing haplotypes can detect parental contribution for germline mutation. We have one option, A and D. So germline mutations are the mutations present in the DNA of germ cells, while the somatic mutations are present in the somatic cells. So we have one option which is A and D, which is true. Germline variants can be inherited from both one or both the parents because these mutations are present in the germ cells. So they can be inherited from parents to the offspring, while the somatic variants they are acquired during the individual lifetime. They somatic cells they cannot be inherited from parents to the offspring, and so these mutations they are acquired during the lifetime. We don't get it from our parents. We acquire them during our lifetime. So the A option is correct. B is somatic variants are always pathogenic while germ-like variants are always benign, which is not the case. So we have seen there are synonymous mutations which can occur in both the somatic as well as germ-line variants. So in both the cases they are not pathogenic. So uh, this option is wrong. C is somatic variants can be passed on to offspring while germline variant cannot be inherited, which is not true. And because germline variant are present in the germ cell, which is inherited from parents to the offspring, not the somatic variants. D is phasing haplotypes can be detected parental contribution of the germline mutation. So phasing means that we are differentiating between the mother and the father chromosome. So this phasing haplotype it can detect parental contribution for the germline mutation, which is a true statement. So the correct option for this question would be A and D. So I hope this is clear. So that was our last question for today's session. So if you have any doubt for today's session. I hope these please go through the manuals that I have shared, so they will help you how to read these files like VCF file or the SAM file. I have not given the manual for the SAM file, but just you can go through it. Go to any manual, just pick a manual, just go through it. You will know how to read the files that we get after using a certain software for doing our analysis. So you will be able to, like, if you go through it multiple times, you will be able to like read them properly. So just practice, and I hope uh, you understood today's session. So if there are no doubt, uh, yes, Deepak.
Ma'am, I have some doubt regarding the exams. Like our final exams, ma'am, I have not. This is my first SOYAM course, so I don't know the level of questions that come in the final exams. So, can you please give a little bit information about it?
So, okay, so I am also not aware the level of the question they will give because we don't make the question, but these assignment, the quizzes you are getting and the assignments we are solving, the question would be like this only, and like you will be able to answer those questions if you do the regular quiz and attend all the problem solving session as well. So I'm not aware of the level of the exam, but they would not be much different from what we are discussing here.
Thank you, ma'am.
Yes. So are there any other doubts? So if there are no doubts, we can end our session. So thank you all for joining. We'll again meet the next week at the same time for our next problem solving session. Okay, so you guys can leave the session. Thank you for joining in the session.